We have created an insulin-resistant fly, which is the hallmark of type 2 diabetes. We took the fruit fly model because it's been used by researchers for over 100 years. We essentially know the fruit fly inside and out.
So what we wanted to do is to develop a model that may help us understand the development of metabolic diseases, age-related metabolic diseases such as type 2 diabetes. We fed the fruit flies 4 different diets, healthy diets here and left, and then overfeeding diets that would simulate overeating like we could see in mammals or humans actually. So when we split these two overeating diets in either overfeeding sucrose or yeast extract, we see that these flies become obese and on both overfeeding parameters, on both overfeeding diets, and develop metabolic abnormalities. For example, flies overfed the sucrose, the carbohydrate source, become obese and develop increased fat storage. Interestingly, when you overfeed these flies the protein source, you see the exactly opposite effect. Fat storage goes down. This antagonistic relationship between these two macronutrient sources continues when we, for example, measure fertility. Flies overfed the sucrose, the carbohydrate source, have much lower fertility as determined by egg counting than flies overfed the protein source. They have much higher fertility. When flies are overfed both of these diets, their lifespan decreases massively. So overfeeding, while having subtle differences on overall metabolism of these flies, definitely leads to a shortening of lifespan. When we use our assay to determine whether these flies are insulin resistant or not, we see that they clearly are developing insulin resistance on both overfeeding diets. It's a little bit stronger on the protein overfeeding diet, but in both cases they develop insulin resistance. So now that we have flies that develop insulin resistance in response to these nutritional stimuli, we have a fantastic tool for further discovery research. After dissecting the fat cells out from the fly, from the adult fly, we stain them and now look at them under a fluorescent microscope. And then we see images like this. We have an assay that will let us determine the strength of insulin signaling. So here you see adult fat cells of a fly stained with a blue dye that stains DNA, and therefore this depicts the nuclei, and a green dye, which is our reporter dye for insulin signaling activity. When insulin signaling is active, this reporter dye gives this very nice, almost cobweb-like staining pattern. It is then present in the plasma membrane, and this gives you this cobweb-like staining pattern. So in this case, this clearly depicts fat cells in which insulin signaling is very active. When insulin signaling is not active, you can see here the reporter is not anymore in the plasma membrane, but you see this diffuse staining in the cytosol, and that is the indicator for us to determine, to label this kind of occurrence here as non-active in insulin signaling.
And the hope is that this will be a useful tool in the future in order to do drug assays, in order to characterize genetic mechanisms of diabetes, just to have a better overall understanding of what makes type 2 diabetes type 2 diabetes.